Did you know that according to the World Health Organization, over 268 million people globally suffer from depression, and this cuts across all ages? You will agree with me that like attracts like, so an organic problem should be best solved using an organic solution. Human and planetary health, indeed, everything is connected. Gomphocarpus fruticocos is an indigenous South African plant that is traditionally used to treat depression-like symptoms. What then is the connection between Gomphocarpus fruticocos and depression at the morphological level, the histochemical, the elemental, and the total phenolic level? I am Akwu Nneka Augustina, a postdoctoral research fellow at the Northwest University, South Africa. How then did we go about this research? Phytometabolites, like you all agree with me, because of their presence in plants, plants are being used for therapeutic applications such as depression and others. Besides the phytometabolites present in them, most importantly, the secondary metabolites, the mineral elements in them also play a role in their therapeutic use. How then did we analyze the micromorphology, the histochemistry, the elemental, and the total phenolic content of this plant? For the micromorphology, we used the fresh leaf samples and fixed it in 70% ethanol according to the description, the protocol, the procedure given by Kim 2011, Kim et al. 2011. For the histochemistry, we carried it out using fresh plant materials, sectioning and staining with different stains, and we observed using the bright field and also using the autofluorescence. For the elemental, we used the powder form, and we followed the procedure described by Magu et al. 2009. For the total phenolic contents, the powder, we use different solvents, the aqueous and organic solvents, 6 organic solvents, to extract the phytometabolites present in them. And we analyzed the total phenolic content using the following Keokatsu reagents assay. And what would we arrive at? We discovered that for the scanning electron microscopy, to study the micromorphology, there are 2 types of trichomes of this plant, the glandular and the non-glandular trichomes, as seen in figure 1A. The glandular trichomes house, they secrete, and then they store these phytometabolites that are important for this therapeutic application. And also, we also noticed that there were also latex on the surface, hence the plant is called milkweed. We also analyzed and observed that the stages of development, that's for the emergent, the young, and for the mature leaves, the distribution and density of trichome vary, which is age dependent. More so, we observed that the older leaves had more of striations on the epidermal cells, the cortical striation. This goes to show, and also most of the trichomes on the mature leaves had already expelled or released their metabolites, and they were no longer metabolically active. It might be deduced that the younger leaves should be preferred for analysis because they have more of these metabolites and stored up and in a large quantity. The figure 1H shows the latex present in the cells. For the histochemistry was done to see where these phytometabolites of therapeutic importance are present and localized within the cells of the plant. And we observed, as seen in figure 2A, that the glandular trichomes GT is rich in alkaloids using the Wagner's reagents test. We also observed that for the UV light, that's figure 2E, that the plant is rich in lignin. You will observe the dark area in there. It's because the plant is rich in latex. So the latex kind of absorbs the UV, so it does not reflect it out. Now for the mineral elements composition, we observed that the plants, because of the presence of the latex, we also analyzed the latex, the composition of the mineral elements. And we observed that the plant is rich in carbon, oxygen, and more importantly, it has magnesium, silicon, potassium, calcium. And these mineral elements have been proven to exert antidepressant effects. Furthermore, we observed that for the phenolic content, the methanol extract gave the highest yield of phenolic content. However, it is important to know that there was a positive correlation between the extract and between the phenolic yield and the extract. So from all of these results, we can deduce that indeed, Gomphocarpus fruticocos is an indigenous medicinal plant that is important for the treatment of